Hello everybody and welcome back to part 2 of exam 3 for PPI. And just a little bit of a disclaimer, I know your first exam was a little odd, and this is the first video I'm recording since your first exam. I know Dr. Weber has changed up the curriculum this year. For reference, I am class of 2026. I was taking this course when Dr. Godelsky was still teaching it, and since he retired at the end of 2022, I know Dr. Weber is the course director now, and I know he is, from my understanding, reusing Dr. Godelsky's old exam content, but I know he's teaching it differently in class. So, at least that was the case for the first exam, and I know that was maybe a little controversial. So there's a chance that if the first video was helpful to you, these will continue to be helpful unless he decides to write his own exams, in which case I still think these are valuable. I just don't know if they would be as one-to-one as the first video. So I apologize in advance if that's the case, because I do not have access to the new content. But with all that out of the way, let's jump into it. So this video I wanted to cover just kind of the basics of neurotransmission. So let's just talk about the basic aspects of that. So nerve cells, they're pretty complicated, but there's really only 3 parts you need to understand for the purposes of this class. You need to understand the axon, the nucleus, and the dendrites. The axon is kind of the functional unit of a neuron, right? This is what sends the signal from the nucleus, whatever the nucleus determines the signal to be, down, and it kind of sends it where it needs to go, down this long chain. And that chain is called the axon. And then here you get to something called the synapse. This is kind of the space in between neurons. That's all it is. It's not a specific part of a neuron, but rather the space in between them. What happens at these synapses is the axon will synapse with what are called dendrites. Dendrites are these long branch-like structures. They have a high surface area. And the reason for that is because neurotransmitters are released at the synapse. So the dendrites will try to scoop as many of them up in order to receive the signal, which they then transmit that to the nucleus, which can choose to make another signal or make another response of some kind, or maybe blunt a response. And that's essentially how nerve cells work at a basic level. So there's a few places where axons can actually synapse traditionally in most cases, and especially for the purposes of this class. You will see axons synapsing to dendrites. However, they can also synapse to neuron bodies as well as other axons. So that's just another type of signaling. And yeah, I mean, you don't need to know that too much in depth, but I just thought I'd mention it. So retrograde signaling is a type of signaling that is a little untraditional. Essentially, the idea is so I'll write retrograde down. It's like a neurotransmitter will either upregulate or downregulate itself. And how does that work? Essentially what it is you can see in this example, we have nitric oxide being released from this end of an axon, right, into this synapse. So it crosses over here into this other neuron. And you can see here, this growth factor, this NGF, is actually stimulated by this nitrous oxide, which will then go back into the original axon, back up into the nucleus, and make a new signal. And usually I would say this is negative feedback. This is actually an example of positive feedback with this NO-NGF pathway. More nitric oxide will be released, but just knowing there's both. Usually neurotransmitters will be regulated or regulate other neurotransmitters, but in some cases they can regulate themselves. So that's just kind of an interesting example that you might be asked about. So essentially, where do these neurotransmitters come from? They're stored in something called synaptic vesicles, or just vesicles. And essentially what they are is these are almost like little bubbles inside of cells, inside of the axons, that will contain just whatever the neurotransmitter is, whether that be acetylcholine, whether that be nitric oxide, which we just talked about. And when something like calcium Ca plus 2. This is probably the most common thing that will release vesicles. This also stimulates like insulin release, so it also works with hormones. Um, when this signal activates, these vesicles will move to the surface, and they'll essentially just dump everything that's inside them into the synapse. And this will obviously allow it to go to the other side, or in the case of retrograde, go back in to make a kind of signal happen. Um, yeah. So the one last thing I want to talk about on this slide is there's essentially multiple clefts. Um, yeah, so this is important. Multiple clefts, which can determine the strength of a signal. So let's say this area right here, right? These all these black dots and also these blue ones. These are actually the same neurotransmitter. Um, there's multiple kind of spaces they can get into, depending on how much they are, right? So if this is primarily releasing from the middle of this, it'll go into something called the primary cleft. This is where the signal is going to be like the strongest. Um, Generally, it's like a very wide available space. You might also have something called secondary clefts. I don't love this image because in this image, the secondary cleft looks bigger than the primary cleft, which is almost never the case. But the point of the secondary cleft is like you have so many molecules that they're spilling over from the primary into the secondary, which can produce an even stronger and possibly different response depending on the type of receptors that are in the secondary cleft. So that's just another basic concept to know. Okay, so now let's talk about action potentials. You might have seen this in like Anatomy 1, or maybe you took Psych. I know it also pops up there as well sometimes. And essentially an action potential is a type of signal that still will use neurotransmitters in most cases. But when neurotransmitters are released, it's more because of like an electrical signal. Um, so how does this work? So essentially, an electrical signal will travel down the axon, which is kind of how they work, right? So even in some of the examples we've seen so far, these might be action potentials. Um, and they will usually activate calcium receptors, which we talked about. This electrical signal might open calcium channels. And calcium isn't in the cell by default too much. So essentially when those channels are open, the calcium will rush into the cell, and then it will cause those vesicles to fuse to the membrane like we talked about before, and release their neurotransmitters into the synaptic cleft, which we alluded to. We just didn't necessarily say that was because of an action potential. Um, so we've been talking a lot about the kind of signaling neuron, but not so much the receiving neurons up until this point. So I wanted to start transitioning into that. Um, and different types of receptors for neurotransmitters. This first one is called an ionotropic or inotropic receptor. I've seen it written as both. I don't know which one's technically correct. I think it's inotropic, but I think this description might have spelled it wrong, but that's okay. Um, Essentially what will happen is a neurotransmitter will be in the cleft and it will essentially it can move through a channel. Um, But you can see that on the side the channel is kind of closed off, right? So how do we open that channel? So we can have a different signaling molecule, or in this case, I think I've got it backwards. I apologize. Um, so this can actually be for both. An ion could bind an inotropic receptor and let a neurotransmitter through, uh, or alternatively, a this is, I feel, a little more common. A neurotransmitter can bind the receptor, right? It's closed off here, nothing can pass. It will open up and allow these ions to move through. And I kind of just alluded to that a little bit. It wasn't an inotropic receptor, but the electrical signal allowed calcium channels to open. So that's kind of an example of that. And there's a lot of subsets of this. It's not incredibly important to understand it all, but just the basic that a neurotransmitter can open up an ion channel. Um, there's another type of receptors called G protein coupled receptors. These are a little more complicated in concept, but not really. Um, all it is a neurotransmitter will bind a G coupled protein. And essentially what that means is two things. A G protein is a protein in the membrane of a cell. This is like a cell membrane. This can also be like the mitochondrial membrane. It doesn't really matter. And these might exist in the nuclear membrane. I actually don't know if they do or not, off the top of my head, but just in some kind of membrane, in like a biological kind of a cell or mitochondria, right? Um, and this yellow protein will actually originally be linked to this. I'll draw this in yellow if that's a little more easy to see. Yeah, so this is right here. But when a neurotransmitter binds, what'll happen is it'll actually kind of break off. Um, and then it could do a number of things. But usually what it'll do is it'll bind to another protein. So now you can see it moves over here and goes onto this channel. Well, when this protein's on this channel, it actually opens up the channel. So you can see you're just opening up the channel. There's basically just one extra step, and that's that a protein needs to break away. Um, and bind to another thing. Another example of this same thing, a neurotransmitter might break this protein off, but it might instead bind to something like an enzyme to either activate it or inactivate it. So there's enzymes in the nucleus, or I'm sorry, not the nucleus, the cell membrane of cells. These can cleave some molecules to make them more ready for absorption. These may destroy waste. So there's like a number of things they can do. Um, and in this case, it looks like this maybe activates the enzyme in this case. Or there's even something called secondary messengers, which can also occur. Uh, just a note that this type of receptor up here can also have a secondary messenger. But essentially all that is it's something like a very common one is adenosine monophosphate. You might recognize that. It's very similar to ATP. This is just, there's one phosphate group. This is just like an energy carrying molecule, which can lead to a cascade of other effects in the cell. This is called the signaling cascade. It's not super important for this, but just something to be aware of. Um, Something else are what are common neurotransmitters? Um, I'd say the most common ones are catecholamines. You have dopamine, which we won't talk about too much in this class, but norepinephrine we definitely will. This is kind of our primary sympathetic catecholamine. And then we also have glutamate, which is excitatory, and GABA, which is inhibitory. You might get a question on that. But these are amino acids. Our primary parasympathetic um, neurotransmitter is acetylcholine. And this is also one we will go pretty far in depth on when we talk about the parasympathetic systems. Um, so now that we know the neurotransmitters, how do these kind of affect the body? So catecholamines in the sympathetic nervous system will traditionally be known as like our fight or flight uh, neurotransmitters. So some things you can imagine, you will have a higher heart rate, you will like breathe a lot faster. Um, your blood pressure will go up, your heart rate will go up, your eyes will dilate. Parasympathetic is kind of the opposite generally. It is your rest and digest system. Um, Basically opposites of the things I just said. Heart rate will slow down, your breathing will slow, your GI tract will digest more. You might need to urinate or defecate more just because in a fight or flight situation you wouldn't need to do that. Your pupils might constrict, things of that nature. Just you don't need to be in a super high energy mode. Um, yeah, and that's at a very basic level how those work. I'll have an entire video talking about sympathetic and another video for parasympathetic. So this is the very brief introduction. Um, Next up, I want to talk about two enzymes, right? And these will work on catecholamines. And the reason these are important, right, is because these neurotransmitters were always floating around our body. Especially something like a catecholamine, something that like raises your heart rate and your blood pressure. That would be really bad because that could give you something like a heart attack. So the two common ones are monoamine oxidase and catechol O methyltransferase. So MAO, wow, that's a terrible M. And COMT. That's not much better, but that's okay. And essentially, these will just break down your catecholamines, right? Like your epinephrine, your norepinephrine. Um, you can see here the amine group gets removed for this carboxylic acid. And you can see here this hydroxyl gets changed to an ether. I don't know if they're going to specifically ask you. They might ask you about catechol O-methyltransferase because I don't think this mechanism is always standard. I don't think it's always a carboxylic acid, but I think this one always makes an ether. So know for COMT that this makes like an ether, or if nothing else, that it will like change a hydroxyl group on that catecholamine. And we'll talk a little bit more about why these are called catecholamines in our sympathetic video. And we'll also come back to these enzymes. So if you don't quite understand it now, don't worry too much about it. This is more of an introduction. Okay, so like we've kind of alluded to so far, is that channels can actually reuptake their own neurotransmitters, right? Whenever we release a neurotransmitter, it wouldn't be efficient for our body to kind of just waste it. So we'll essentially have like channels that can reuptake it. You might have heard of SSRIs. These are probably the most common psychiatric medications these days for treating anxiety and depression. Um, these are selective serotonin reuptake inhibitors. So these work on the SERT channels, and essentially what they are is they will prevent serotonin from being reuptaken into the cell they were released from, and this will allow it to stay in the synapse longer and release the secondary cells, which will actually regulate like mood and ideally have a positive effect. Although in some people it's negative. Serotonin-based medications are strange, and psych medications in general. But anyways, yeah. So DAT, NET, and SERT are three very common reuptake channels. I wouldn't expect you to know that for the exam. Maybe CERT, but don't worry about it. But what I would expect you to know is, like we mentioned before with selective serotonin reuptake inhibitors, there are other ones, and two very common ones are drugs of abuse, amphetamines and cocaine. Um, and these prevent norepinephrine reuptake as well as I think dopamine reuptake. They each have a slightly different mechanism. We will talk about them in our sympathetic video a little more in depth, but just be aware that these can block this reuptake. And in the case of a sympathetic neurotransmitter like norepinephrine, more being in the synapse, that's a very like excitatory response and causes a lot of stimulation, high heart rate, high blood pressure. So that's why you hear people taking things like cocaine will have heart attacks. It's not so much the cocaine itself, but rather there's so much norepinephrine flooded into your system that it's causing this giant sympathetic effect. Um, Acetylcholine, on the other hand, if you remember, this was our parasympathetic neurotransmitter of choice. There is an enzyme that breaks it down. These are called acetylcholineesterases. This is kind of like the equivalent for acetylcholine that something like COMT would have been, or MAO. Um, ACHE is the most common way acetylcholine is broken down by far. Like to the point where it's almost exclusively stopped this way. So we have certain drugs that are called acetylcholinesterase inhibitors that will block this enzyme. And essentially what that does is that'll make more acetylcholine and we'll have more of that parasympathetic effect, right? More of those relaxing effect, low heart rate, uh, low breath rate. And that could be good. We don't really use them for blood pressure. We can use them for like Alzheimer's and dementia actually, and also for glaucoma. So kind of not things you'd expect. I think we already talked about their use in glaucoma actually. And yeah, they're also used in some mental disorders, some very niche ones. But just kind of interesting. So yeah, that's basically, oh, I even say it right here. That's funny. Sorry, I was looking at my notes. Yeah, so it's used in like Alzheimer's. This is called the cholinergic hypothesis. Might be a question, but essentially the idea behind it is that loss of cholinergic neurons in the brain uh, is kind of the pathophysiology of Alzheimer's. Again, we don't really know because the brain is so complex. Um, but this is the FDA approved treatment for Alzheimer's, which is kind of interesting. I think it's the first line treatment. So yeah, kind of surprising. So next up, I want to talk about kind of how the nervous system is broken up. There's two main parts, but then there's a lot of subsections as well. The two main parts you need to know are the central nervous system and the peripheral nervous system. Central nervous system, this is your brain and spinal cord. Um, this system is kind of like isolated by itself, which kind of makes sense, right? We don't need our brain per se, like controlling all of our body at one time. It's more important our brain regulates itself, and specifically, like you might have heard of the 12 facial nerves from anatomy, including like your optic nerve, your glossopharyngeal nerve. There's a lot of them, vagus nerve. Um, these very essential sensory organs are what they're all part of the central nervous system because they're so important. And then also, it's a little surprising, but the diaphragm, the muscle that allows you to breathe, is also controlled by the central nervous system. Most of our body, on the other hand, is controlled by the peripheral nervous system. Um, and there's two subsections to this. And then one of them also has a subsection. There's somatic. So somatic is our skeletal muscle. Basically, the way I think of it is if you can move it, it's probably somatic. Like me clenching my hand into a fist or moving my arm. This is all part of the somatic nervous system. The nerves I'm using they're part of the peripheral nervous system, and then subdivided into somatic. Uh, and they control skeletal muscle, which skeletal muscle is the kind of muscle you can control. Autonomic is primarily smooth muscle. These are things you cannot control. For example, I can't like change my heart rate at will. I mean, I guess I could go on a run and make it faster, but that's not really me changing it. That's the fact that my body recognizes that I need to pump more blood. And this is broken into those two categories we've been alluding to, that's sympathetic and parasympathetic. Um, we'll talk a little bit about somatic um, in this module, but not a ton, and we pretty much won't even touch on CNS at all. But just something to be aware of, because that'll come back eventually in therapeutics. Um, so the way that I kind of like to think about these is because they all work a little bit differently. You have your somatic nervous system, right? This will just innervate a skeletal muscle. There'll be one long, what's called, well, there'll be one ganglia, kind of this terminal, um, as well as one real neuron. When it comes to the autonomic nervous system, you can see there's actually, in both the parasympathetic and sympathetic, there's actually two neurons for each, right? There's like a primary and a secondary. Um, or sorry, they put one in the middle. It's these two right here. Um, and you can see they're like different length. Like this one has a really short neuron at first. This one has a longer first neuron. Then it's the opposite. Parasympathetic has a shorter secondary ganglia, but a longer like primary one, and it's opposite for sympathetic. Something to note. I don't think that'll be a question. And then there's also like other types, but I would say know these three and know that this is para and this is sympathetic. That would probably be the biggest thing I'd take away from this. And like I've alluded to a few times now, sympathetic, right? Blood flow, bronchioles are dilated. Those are structures in your lungs. They help you breathe more. Liver releases glucose. The reason this happens is essentially because if you're in a fight or flight situation, you need energy. The liver is kind of like your glucose store. Um, and it'll just release glucose to power your body. And then blood pressure increases. And then parasympathetic, the opposite, right? Low blood pressure, high GI activity, you're digesting, and pupils get constricted. So, something I've been talking about kind of is some organs will be innervated by both parasympathetic and sympathetic. A really good example of this is like your heart, right? Um, M2 receptors, which you don't need to memorize this because we'll talk about it a little bit more in their individualized videos, will slow down the heart when they're like hit by acetylcholine, right? That's a parasympathetic response. Versus like beta 1 receptors will increase heart rate. That's sympathetic response. It's stimulated by catecholamines. Um, Another example is the GI tract. Uh, we have like what's called the myenteric plexus. You won't need to know this, but it's essentially there's some signals that will increase secretions, and enzyme production, things of that nature. And there's some that will, the sympathetic response will kind of slow down the muscles that help move, or I'm sorry, the muscles that help move things through the GI tract. This is a pretty complex photo. It was more just to show how many factors are controlled by these systems, but I almost regret throwing it in here because even now as I'm looking at it, this is a very complex image, and I really should have stuck to my heart example. So just focus on the heart. Just know that multiple things can innervate other multiple things. We alluded to this on the previous slide, right? Sympathetic have, when it comes to the length of their the ganglia of their neurons, they have a short presynaptic and long postsynaptic if they're sympathetic. Parasympathetic is the opposite, long pre, short post. Um, and then their ganglia is kind of like where the pre and post ganglionic neurons meet. It's the space in the middle. So right, if there's a short pre, it's obviously going to be close to the spinal cord because it didn't travel that far yet. And then if it's a long pre, it's going to be close to the actual organ it's working on. So that's just kind of the idea behind that. I included this here. I don't think you need to know the origin of the fiber, um, but it's there for your reference in case you were interested. And then something else I did want to mention is your preganglionic neurons will usually have a lot of what's called myelin on them. Myelin is something that wraps around the axon. It's like a protein, and it essentially just allows the signal to transfer faster. Um, and you can see that the preganglionic neurons are myelinated, right? We want that initial signal to be really fast, but then our postganglionic, we just don't innervate. It's not incredibly necessary to get it to the secondary location as fast. Granted, these are still happening in like a matter of like milliseconds generally. Uh, but I'd say myelinated from what I remember from psychology years ago is it's about 5 times faster. I could be wrong with that number, if anyone wants to correct me in the comments. But yeah, just be aware of myelin. You might get a question on it. Uh, so when it comes to sympathetic responses, uh, and this is a mnemonic Dr. Udelsky used. I'm not a huge fan of mnemonics, but I'll put it down here. He called them E situations. And by that he meant like exercise, um, emergency, excitement, and embarrassment. So what did he really mean by this? Essentially, this is part of your sympathetic nervous system, right? So what happens when you have these? And we'll talk about these more in our individualized videos. But essentially, if you get really scared, you might notice that your pupils kind of dilate, or you might have seen like before that, like if you, the common scenario I've seen it in is like your pupils dilate if you're attracted to somebody a little bit, that kind of excitement. It's your sympathetic nervous system that's doing that. Same thing. If you're scared, your heart rate will go up. That's just to pump more blood. Um, Pretty common. Uh, we have something in our lungs called bronchi. These are essentially little, I almost think of them like valves at how much air we can get in and out of our lungs. And there's like millions of these. So it's not like one central valve, but essentially they will dilate in an excited state because if we're trying to like escape an emergency, we need more air to deliver more oxygen. So that's why they do that. Um, Next thing is blood vessels in like the GI tract will actually constrict. Um, the reason for this is in an emergency situation, we don't need to be digesting. We need to get that blood to our skeletal muscles. So what'll happen is, I don't know if there's a picture around it around here on this diagram, but yeah, I guess there's not. Okay, so that's something I can draw in. So pretend this is a skeletal muscle. Um, these vessels will actually dilate, whereas your GI tract would constrict. The reason for this is if we're running away, right? We need oxygen to be moving to like our legs if we're running. So that's why they dilate. Liver will release glucose. We alluded to that just as like an energy store. Um, and then the abbreviation Dr. Godelski used for parasympathetic was SLUD. It stands for salivation, right? And this kind of goes back to the idea of digestion. Your saliva has digestive enzymes, even if we don't think about that too often. So if you're in a non-emergency state, you will salivate more. And you could even think of this not as like non-emergency, but when you're sitting down to have a meal, you produce more saliva. Same kind of thing. Lacrimation, which is tear formation, you'll have more of that. And that's not so much you're crying, but more just like your eyes are producing more fluid to keep them like just in a regulatory state. Parasympathetic's more of a, like a regulatory kind of system. There's urination as well, right? You're not going to go to the bathroom if you're running away from something, but if you're just bored at home, you might. Digestion, I've alluded to this, more digestive enzymes in like your GI tract. And then there's, for the final one, it is defecation, right? So again, going to the bathroom. Um, yeah, so pretty standard, like, I wouldn't necessarily call them relaxing activities, but things that happen when you're at rest. And then also, uh, he mentioned the 3 decreases in class. You have decreased heart rate, right? Kind of the opposite. Decreased bronchi dilation. So they'll be more constricted, right? That valve will be more closed. You won't be delivering as much oxygen. And then also a decrease in the diameter of the pupils. You're not letting as much light in. You don't need to see as much. And that's kind of why that occurs. So, something I want to talk about now is kind of the receptors of the parasympathetic and sympathetic. Uh, the videos we have coming up on those are very comprehensive, but I would say this is just a really good diagram to kind of look at these and recognize these. So there's two kinds of molecules that could hit any of these receptors, right? They're what are called agonist and antagonist. Um, And we've talked about this in our first exam. Remember, agonist will upregulate a receptor. They'll have it produce its response. Antagonist will not upregulate a receptor. They also won't downregulate it. So for example, let's say we are hitting a beta 1 receptor, right? This is a sympathetic receptor that increases heart rate when it's hit by an agonist. An antagonist won't lower heart rate. That would be what an inverse agonist does. An antagonist will just prevent a heart rate from being raised. So let's say at baseline your heart rate is 80 BPM. An agonist might make that like 120 BPM, right? An antagonist would keep it at 80 BPM and essentially not allow, even if we had a ton of like norepinephrine, right? A ton of sympathetic signaling molecules, especially if it's an irreversible antagonist, it would just make it so your heart rate doesn't go above 80. So that's kind of where that works. You can see here, the sympathetic and muscarinic, they're or sympathetic and parasympathetic, I'm sorry. I also call parasympathetic muscarinic sometimes, so if you ever hear me do that, just know that's what I'm talking about. And I might also call this adrenergic for sympathetic, and that's honestly a good thing for you to know as well. They're interchangeable terms, but just be aware of them. Parasympathetic's pretty easy. Heart has M2 receptors, and literally everything else is M3. So if you're ever on a test question, it's asking what kind of parasympathetic receptor is this, M3 is almost always a safe bet, unless it's the heart, in which case it's M2. Adrenergic is unfortunately a little bit more difficult. You can see there's 2 types of receptors. There's alpha receptors and also beta receptors. These will vary a little bit. I won't go too in depth on these right now, just because again I have like a whole video explaining what these receptors do and where they work. But what I will say is that norepinephrine works on this side, acetylcholine works on this side, And then, right, these are their agonists. There are antagonists. We'll discuss those more in the videos. Antagonists to these in the body are usually actually just drugs. For example, a beta antagonist might be used to lower heart rate, and also it could constrict airways, which isn't ideal. But that's kind of like what it would do. Versus like an agonist would increase heart rate and increase those blood vessels, which would be norepinephrine in this case. So our last thing about autonomic nervous system is that there's something called reflex arcs, and you might recognize these from psych or from anatomy. Uh, these are essentially like self-regulating, either negative or positive feedback things, and these just maintain homeostasis. Um, so for example, there's a lot of ways we can modulate our own blood pressure in our body because our body wants our blood pressure to be at a certain level. We can adjust heart rate, force of contraction, or vessel diameter. This is an equation you'll talk more about in therapeutics, but it's blood pressure equals heart rate times resistance. You might have also seen this in anatomy so far, but heart rate is essentially like how many beats per minute your heart does. Resistance is a little more vague, but think of it as like the diameter of your vessels. And if the diameter of a vessel is smaller, uh, resistance is higher, right? Because it's kind of like, let's say you have like a hose and you constrict it. The water that's coming out is going to be like much more pressurized, and it's the same kind of thing. So that would increase your blood pressure. So basically, we have something called baroreceptors in our circulatory system, which it doesn't really matter what this is called. It's the vasomotor center. It's in your brain. That's not super important. But just knowing, let's say you have a heart rate, and for some reason your body just cannot get it down, and you have high blood pressure right now, you have a high heart rate. How your body would respond is it would change the venous tone. And in this case, it would dilate your blood vessels. I don't know if that's what this diagram is showing. I'm kind of making up my own example. But essentially, right? Because you want to keep that blood pressure lower. And since this is a multiplication equation, if you have a high heart rate, the thing to lower that would be to have a low resistance. And to do that, you would dilate your blood vessels. And we'll talk more about that in the sympathetic video. So if that doesn't make sense right now, don't worry too much about it. Um, and these are all the specific names of the centers. Effectors are kind of like the actual thing that's changing. Um, these could be like, in the case of our example I just gave, the way blood vessels change size is by smooth muscle. Um, if we'd done an example where instead the resistance could have changed, the heart rate would have changed, so we would use cardiac muscle. Also, even glands, right? Like glands can produce neurotransmitters, so we could just do that as well. Like if we wanted that to go down, we could release acetylcholine for like a muscarinic response. But with that, I think that's everything on neurotransmission. Thank you so much, and I'll see you in the next one.